Hey everyone! Welcome back to B.Pharm buddy. If you are new here, don't forget to hit the subscribe button and turn on notifications for simplified pharmacy education and career tips. Today, we are diving into an essential topic in pharmacokinetics, membrane transport mechanisms. So, grab your notes and let's get started. Pharmacokinetics is all about what the body does to a drug, how it's absorbed, distributed, metabolized, and excreted. And the first step of this process, absorption, often depends on how drugs cross biological membranes. But wait, why do drugs even need to cross membranes? Well, most drugs are administered at one site, like the stomach or muscles, but they need to reach their target site in the body. And to do this, they must pass through cell membranes, which act like gatekeepers. Cell membranes are primarily made up of a phospholipid bilayer. Think of it as a sandwich. Phospholipids have a water-loving head and water-fearing tails. These arrange themselves so that the heads face the watery environments inside and outside the cell, while the tails stay tucked in the middle. This unique structure makes the membrane selectively permeable. Only certain substances, like small, nonpolar molecules, can easily cross. So, how do drugs manage to cross this barrier? Let's explore the mechanisms. First up, we have passive transport, the simplest way for a drug to cross. This process doesn't need energy and happens in two main ways. Drugs move from an area of high concentration to low concentration until equilibrium is reached. This works best for small, lipophilic, fat-loving molecules like oxygen and alcohol. Here, drugs still move from high to low concentration, but they need a little help from carrier proteins or channels embedded in the membrane. An example is glucose transport via GLUT proteins. Next, we have active transport. Unlike passive transport, this requires energy in the form of ATP because the drug is moving against the concentration gradient from low to high concentration. Active transport is highly specific and involves carrier proteins. A classic example is the sodium-potassium pump, which maintains ion balance in cells. For drugs, this mechanism is critical for substances like amino acids or glucose derivatives. Now, let's talk about endocytosis. This is when the cell engulfs a drug molecule into a vesicle. Think of it as the cell swallowing the drug. Endocytosis is vital for transporting large molecules like proteins or nanoparticles. There's also specialized transport mechanisms like ion channels for charged molecules or gap junctions for direct cell-to-cell movement. So, what determines how easily a drug can cross a membrane? Here are some key factors. Smaller molecules cross more easily. Lipophilic drugs pass through membranes faster. Ionized, charged drugs struggle to cross. Non-ionized ones are absorbed better. The pH of the environment and the drug's pK influence its ionization. Understanding these factors helps pharmacists and researchers design drugs with optimal absorption properties. Let's quickly recap. Drugs cross membranes via passive transport, active transport, or endocytosis. Passive transport includes simple and facilitated diffusion, while active transport needs energy. Factors like size, solubility, and pH play a major role in determining a drug's ability to cross membranes. Understanding these basics is crucial for mastering pharmacokinetics and excelling in your pharmacy career. That's it for today's video. If you found this helpful, don't forget to like, share, and comment below. Let me know what topics you'd like me to cover next. And as always, subscribe to B.Pharm buddy for more content like this. Until next time, keep learning and stay curious.